Yeah, thanks everyone for allowing me to talk today. I think a lot of the things that Malak talked about in her keynote talk are going to return to the fore. We're going to keep thinking about soil, microbiomes, metabolomes, soil rhizosphere processes, but in the context of carbon mapping and in the context of an agricultural system. Okay. Oh, I had an acknowledgement slide up here, but this sort of came out of a FICUS grant that was in collaboration with a lot of folks at EMSL as well as the Joint Genome Institute. And so what I've been trying to do is sort of integrate all of this together into a nice story thinking about fertilizer use in sorghum agroecosystems in central Arizona. We also are integrating trace gases. So now we're going to start thinking about the gas part of this, which Malak's talk didn't cover too much on, but is an important thing to think about when we're thinking about agricultural systems. Greenhouse gas emissions and nitrous oxide in particular is agriculture is the predominant terrestrial source of that greenhouse gas to the atmosphere. And fertilizer usage is the predominant source of that gas. And so when we're thinking about fertilizer use in agroecosystems, and we're also trying to reduce emissions into the atmosphere, a component of that might be important to think about is what's actually happening in this plant soil in sort of rhizosphere part of the soil where we're adding fertilizer that we eventually want to get into the plants. Plant roots are interacting with microbes in the meantime, and we might be able to manipulate the way that plants and microbes are interacting by using different genotypes of the same crop. So we know we have a lot of cultivars of different crops, and for sorghum, we have this great system in central Arizona where we've actually genotyped, I think, 60 different types of sorghum that differ in their productivity, above ground productivity and fruit productivity. And so we use two different sorghum lines, this low productivity above ground system, which we're calling low productivity based on its above ground growth, and our high productivity sorghum system. And this is a super simplified diagram, but we expect below grounds that maybe if we have a low productivity sorghum above ground, we might have a low productivity system below ground with a smaller root architecture and maybe more condensed microbial community compared to this high productivity system where we might have this larger root architecture, larger root biomass, and more interactions with microbes below ground that might influence fertilizer usage in the soil. As part of this, I also want to show that we're able to actually connect these subsurface probes to actually measure gas production underneath the soil surface on a semi-continuous basis. And we also collected soil cores at different stages of our sorghum growing season. So in Arizona, sorghum gets planted in May and then is harvested in October, September, October. And so we were able to track using these soil cores, different sort of plant growth stages. So moving from where plants were initially planted and don't really have rhizosphere production towards growth maturity into reproductive and senescent stages. Simultaneously, because it's Arizona, which has a monsoon system, we're also experiencing different fertilizer and irrigation regimes. So early in the season, we are adding more fertilizer as fertigation. So that's fertilizer mixed with irrigation together. And then fertigation stops and we're just getting water for the rest of the growing season. So there's a couple of different things at play here. We're looking at changes in plant productive or sort of plant growth stage, climate and irrigation regimes, and different genotypes, and thinking about how all of those might interplay to influence fertilizer use across the growing season. Okay, so I'm going to show you three different types of data, or three different sort of ways that we can approach thinking about this sort of comprehensive mapping of nitrogen within the sorghum system. So the first way is we're going to think about genes and gene activity of soil microbial communities within the sorghum system. And so here I'm just showing you soil metatranscriptome data. So this is actively expressed nitrogen cycling genes by the soil microbial communities. We're not thinking about specific taxa, we're not thinking about groups of microbes at this point, but just what is the overall gene activity, gene expression that's happening in these soils. And so here on the x-axis, I'm showing you, these are sort of faceted by 3 different parts of the growing season. On the x-axis here, I'm showing you combinations of bulk versus rhizosphere soil. So for our August sampling point, we were able to actually separate rhizosphere soil from bulk soil to look at the real impacts of the plant root directly on the soil microbial gene expression. And I'm also showing you different productivity lines. So we had some control plots where no plants were planted, and we had some plots with our low productivity and high productivity sorghum. And here I'm just showing a heat map of 3 different sort of ways, nitrogen pathways that soil microbes will use. So they might be using genes related to organic nitrogen synthesis and degradation pathways. And then these are sort of dissimilatory processes. Nitrification and denitrification are inorganic nitrogen transforming processes that will create our trace gases like nitric and nitrous oxide. What I want you to notice here is that denitrification among the dissimilatory pathways, so that's these top 2 here, seems to be the most actively expressed pathway. This is, I think, not unexpected because sorghum is actually a really cool plant that produces biological nitrification inhibitors by its roots. And so it can actually inhibit this process. And something that I was just thinking about this morning as I was putting my slides like fully together in my head, is that the low productivity line actually seems to have decreased gene activity of some of these nitrification, increased gene activity for particularly this HAO gene. And so maybe nitrification inhibition is not happening as strongly in the low productivity line. In terms of our synthesis and degradation pathways, which might connect up to our metabolomic data that I'll show you in a second, glutamine synthesis seems to be the most actively expressed organic pathway in these soil microbial communities. And glutamine is sort of this key central metabolite for plant growth, microbial growth. And so this also isn't unexpected, but it's nice to see that we have really high upregulation of soil microbial glutamine production. And gene expression in the low productivity lines seems to be lower. If you don't focus specifically on any specific gene, but just look at sort of the striping here, gene expression does seem to be lower in these low productivity soils during plant maturity and reproduction, and especially looking at the bulk soil in particular. But the rhizosphere productivity lines seem to be more similar to each other. So potentially gene activity in the rhizosphere section of the soil might be more similar across plant lines compared to the bulk soil. And then I'm not showing you this here, but we also have amplicon sequencing data to actually look at the microbial taxa. And we see that they do change through time, but don't have a big difference among genotypes. So potentially plant genotypes, sorghum genotypes are not especially influencing the microbial community directly, more the gene activity. So we can also track soil trace gas concentrations of nitric oxide and nitrous oxide here over our 4 different sampling dates and try to get pictures of what is actually being produced and consumed in the soil in terms of gas production. You'll notice later in the season right away when fertilizer, active fertilizer addition stops, we get a plummet in these gas concentrations. And then earlier in the season, when we have fertigation happening or right after fertigation has stopped, we see higher gas concentrations. I don't think that's unexpected, but what's interesting to show is that, or what's interesting to see is that We see predominantly nitrous oxide concentrations being higher than nitric oxide. That might also be expected if biological nitrification is being inhibited in the soils by sorghum. And we do see, nitric oxide concentrations are higher in those low productivity soils. So maybe nitrification inhibition is not happening as strongly in those lines. In July, the low productivity sorghum produces higher nitric oxide, or sort of the soil concentrations are higher in the high productivity lines, whereas nitrous oxide is higher in the high productivity line, which is primarily dominated, I think, here by this initial pulse that's happening right after an irrigation event. But during the latter half of the season, the high productivity soils have lower nitric and nitrous oxide concentrations compared to the low productivity. So maybe nitrogen is being used, I won't say more efficiently, but maybe more of it is being incorporated into the plants or into the soil microbes rather than being lost from those systems. And we can actually start to sort of relate this to gene expression data by doing these comparisons between nitrous oxide production genes and nitrous oxide consumption genes, and relate those to soil nitrous oxide concentrations in the soil. And we do see this very messy, but presumably positively related correlation, where the more nitrous oxide consumption gene expression you're getting in the soil, maybe the lower concentrations of N2O. So that's exciting to see. And then the last part of this this sort of comprehensive mapping, which maybe folks in the room are most excited about, is looking at the metabolome. And so here I'm showing some FTICRMS data. And so when we look just sort of at the overall production of metabolites in the soil and relate those to different compound classes, we see that just the overall richness, so this is the presence of different soil metabolites that are grouped into these specific compound classes. The soil metabolome sort of shrinks across the season. So in the beginning, we have a lot of different types of soil metabolites, and then as the season goes on, we get this sort of constricted metabolome picture. But this shrinkage is particularly high, or particularly noticeable in the low productivity genotype, where we actually have the highest richness initially, but then actually, yeah, if you look at July, we have the highest richness initially, and then it really plummets when we when these genotypes start undergoing reproduction and senescence stages. We can also look at specific metabolites, and mostly amino acids, using this proton NMR data set of just nitrogen containing metabolites. And here in this heat map, where I'm showing you different productivity lines across our different parts of the growing season, we do see that July is a particularly strong sort of higher concentrations are happening for particular amino acids and other metabolites, especially in the low productivity line, and that sort of disappears later in the season. So this is happening in the soil. Conversely, in the roots, where instead of seeing this low productivity crash later in the season, we actually see in the roots that we have higher concentrations of a lot of these specific amino acids and other nitrogen containing metabolites. This is also proton NMR data. Yeah, so we might be seeing more sort of concentration of nitrogen within roots rather than soils in the low productivity lines. And here I'm starting to try to connect this to some MALDI mass spec imaging data, similar to what Malak had shown in her talk. And so we can try to start piecing apart like the spatial distribution of those metabolites within the roots themselves. I initially wrote like potentially there's maybe some exudation happening from the roots into the soil, which might explain why we see concentration of a lot of these sort of nitrogen containing metabolites around the edges. So this is the endodermis here because sorghum undergoes cortex sloughing later in the season. And so we actually don't have a cortex or an epidermis, but the endodermis here does have a high concentration of a lot of these amino acids, or in particular regions of the vasculature and near the endodermis. And so we're trying to start thinking about, maybe there's these metabolites are actually being exuded outside, or they're being used more for structural defense as the roots age and as the plants age. Okay, so I know I talked for a while, but this data is still very preliminary. So I love to have a chat with anyone about sort of how we can start to think about putting these data together more. Some preliminary, but we are starting to see some preliminary patterns, maybe that high productivity sorghum has this larger sort of sphere of influence or rhizosphere on soil nitrogen cycling processes. Even if the roots aren't directly in contact with the soil, we see that the larger sort of rhizosphere of these high productivity lines is resulting in higher nitrogen gene activity, even in the bulk. We see lower nitric and nitrous oxide accumulation when fertigation stops. So this is sort of later into the growing season. And we have this more rich nitrogen containing soil metabolome even later into the growing season instead of the low productivity where we saw a crash. And then this high productivity sorghum might have more dilute N in the tissue. So remember those root NMR concentrations were a lot lower than the low productivity line, which could mean this could be due to potentially more exudation in the soils. We know that amino acids can be exuded into the soils, which might influence soil microbial biomass, or they could just be transported to these above ground structures. And this high productivity line really is better at shunting nitrogen up into its reproductive and growth structures and leaves the roots sort of less concentrated. And I just wanted to leave this up here. I saw this conceptual diagram on Google and in this paper, and was interested in. I think this this particular part of this diagram seems like something where we can really start to use these combined sort of comprehensive data sets to really tackle this part of the question of how to make agroecosystems more sustainable, more nitrogen efficient, and thinking about sort of organic matter accumulation and better usage of nitrogen by plants and soil biomass. So thank you so much. I'll take any questions.
Thank you, Holly. I guess, okay. Thank you, Holly, for this great talk. Do we have time, I think, for one or two quick questions? Okay, here.
No, I feel like after that pause, waiting for the microphone to come, I need to ask a really profound question. So let's see. Yeah, I'm just curious to know, I'm a really interesting kind of shift in mass balance of the dissolved pool of metabolites through the season in the different productivity zones. Just wondering if you've thought about or even tried to measure the exchange with the soil minerals. How much of that nitrogen pool is interacting with the soil minerals? What are they taking up? What are they releasing? Presumably that's going to change in the presence of fertigation versus irrigation because of salinity changes in the soils and just the dynamics of the mineral water interface. So just curious to know if you've thought about how that interacts with the signal you're observing.
Yeah, that's a great question. We haven't really explored sort of the mineral associated versus particulate associated organic matter. We do have sort of water data, so we can relate this to hydrologic cycling and soil moisture and soil temperature. We also have some hyperspectral imaging data that I'm working on processing now that we can try to get at the what is happening above ground, because we didn't take any samples above ground, but we can use these sort of leaf nitrogen indices to maybe estimate whether how much nitrogen is getting up into the above ground biomass of the plants. I know that didn't exactly answer your question, but yeah, so we haven't looked a ton at mineral relations with our organic matter pools, but I think that would be a great next step. And yeah, I would love to talk more.